All right, I think it's safe to say we could get started. Good evening, everyone. Welcome to the 2025 Lupus Ontario Symposium, The Spectrum Within, Intersections of Skin, Sex, and Systemic Change. I'm Julian Capelli, board member and chair of the Education and Awareness Committee here at Lupus Ontario. We're thrilled to kick off part one of this two-night symposium running tonight and tomorrow, and we thank you all for being here this evening. Lupus Ontario is the largest provincial voluntary organization dedicated to improving the lives of people living with lupus. Our mission is to provide vital support, education, awareness, advocacy, and research through fundraising efforts of our staff and volunteer community to help those with lupus live longer, healthier, and better lives. I'd also like to take a moment to thank Roche, our presenting sponsor for this year's Lupus Ontario Symposium. We're very grateful for their generous support, which helps make sessions like this possible. And I'd like to remind everyone that we'll have time to answer questions at the end of the presentation, so please feel free to submit them at any point using the Q&A function or chat box. So let's begin with the first session of the symposium, Heredity and Lupus. Is Lupus Passed Down? With Dr. Joan Wither. And it's just now my honor to briefly introduce Dr. Joan Wither. Dr. Wither is a professor of medicine and immunology at the University of Toronto, senior scientist at the Schroeder Arthritis Institute, and staff rheumatologist at University Health Network, where she is the director of the STAT and Early Autoimmune Rheumatic Disease Clinics. Dr. Wither's translational research program focuses on the systemic autoimmune rheumatic diseases, including SLE, Sjogren's disease, and systemic sclerosis. Her laboratory has advanced the understanding of SLE by dissecting its genetic basis in mouse models and contributing to international GWAS/immunochip studies that identified novel risk loci. More recently, her work has shown how interferons disrupt immune function, identified biomarkers predicting progression from asymptomatic autoimmunity, and uncovered markers that stratify lupus nephritis patients by treatment response and long-term renal outcomes. And now for the presentation. This presentation will take a deep dive into the science behind heredity and autoimmune disease. We'll examine what current research says about genetic risk factors, how environmental triggers play a role, and why lupus doesn't follow a simple pattern of inheritance. Whether you're living with lupus, have family history, or simply curious about how genes and how environment interact, this session offers clear insights and valuable context. So without further ado, I'll now turn it over to you, Dr. Wither. So thank you.
Hey, thank you for that kind introduction. And I'm pleased to have the opportunity to tell you about genetics and the environment in lupus. These are my disclosures. So is there a genetic basis for the development of lupus? And I think this really became first apparent with studies of families where individuals had lupus. And perhaps most definitively through studies of twins. So the twin studies really provide strong evidence that there is a genetic basis for the development of lupus. And on this slide, you can see on the left, we have identical twins who share all of their DNA. And in identical twins, 24 to 69% of the time, if one twin has lupus, the other twin will have lupus. Whereas if you look at non-identical twins, where they only share about 50% of their DNA, only 2 to 9% of the time will they share a diagnosis of lupus. So when they share a diagnosis, this is called concordance. And you can see, though, that it isn't 100% in identical twins. So this means that there must be some other factors other than just genetics that determine whether someone is going to develop lupus or not. And we consider this to be fairly strong evidence that the environment also plays a role. And there have been a number of family studies that have been performed in lupus. And these show that the family members of lupus patients have an increased likelihood of developing lupus. And there's two really nice, large population-based studies where they looked at the first-degree relatives of lupus patients, one from Taiwan and another from Denmark. And what you can see here is that if you have one first-degree relative who has lupus, your overall risk relative to lupus in the general population would be 17-fold higher of developing lupus, or 10-fold higher in this other study of developing lupus as compared to the general population. And this risk is even higher when there are two family members with lupus, where it goes up to 35 to 61-fold increase in the likelihood of developing lupus. And it varies a little bit depending on your relationship to the patient. So if you're the offspring of a lupus patient, then you have about a 14 to 17-fold increased risk of developing a lupus. If you're a parent, it's a little lower. If you're a sibling, it's about the same or slightly higher, where it's 26-fold increase. And this translates to about a 2% risk of developing lupus. And interestingly, because the environment also appears to play a role, the spouses of lupus patients have a fourfold increased risk as compared to the general population of developing lupus. And this, as I said, really provides some evidence that the environment also plays a role. Now, having said this, if you actually overall look at lupus patients, only 1 to 6% of lupus patients will actually have a first-degree relative with disease. So the vast majority of lupus patients develop the disease sort of spontaneously without an obvious family member who has disease. So overall, when you look at these family studies, it's estimated that about 40% of the susceptibility to lupus is inherited. And then the rest of this is from environmental factors. So obviously if you're in the same family, you share many environmental exposures. And it's estimated that of the rest of the risk, about half of it is due to these shared environmental factors, and the other half is due to environmental factors that may not be shared between family members. So in terms of the genetic risk of the disease, as I'm just going to briefly review some very sort of fundamental aspects of genes and DNA. And as you know, our genes are encoded in our DNA, and we have 22 pairs of chromosomes in our cells, with either an additional X and X for a female or X and Y if you're male. And on each of these chromosomes, there's hundreds to thousands of genes. And the gene is really encoded within the DNA. And it's the sequence of the DNA that really tells the gene what it's going to do. And when you look at genes, the purpose really is to encode proteins. So here we have the DNA. It has a specific sequence. It's translated into RNA. And then that specific sequence tells them the RNA what specific amino acids will be comprised in the protein. So these proteins perform all sorts of functions. They perform functions in the cell. They, for example, will convert signals from outside the cell to inside the nucleus. They can be on the surface of the cell, and they can interact with proteins on other cells, so they can allow communication with other cells. They can respond to the environment. So there can be a protein on the surface of the cell, and it can respond to some secreted factor in the environment. So the proteins basically are how the cell function.
One moment, Dr. Wither. Your presentation is small, so is there a way for you to make it bigger? We see mostly you, and then your presentation.
Oh, really?
Yes.
Okay, what I see on my screen is a small me and a big slide. So how can I fix that? I don't know.
Maybe
You
just
want me
to
to,
maybe just try and disable your presentation and then just re
what?
put it back into presentation mode.
I thought it is in presentation mode. Okay, just wait. Stop share. Okay, now what would you like me to do? Share again?
Yep, try once more.
Share. Okay, can you see that?
It's loading, yes.
Is it big?
Yeah.
Okay, now can I put it in slideshow?
Yes, please.
Okay, and then let me go to here and go from current slide. Okay, let's try again. Let's do from the beginning. It's not going to slideshow. Do you see the slide?
Yep, I see it.
But it's not in, I've tried to go from beginning.
When you hit that, nothing happens.
Nothing happens. So here, can we go
Do you see
down
that gray
to
button on the actual screen?
a gray button? Oh, resume slideshow?
Yeah.
Can you see it now?
Yes, I can.
Is it big?
Yeah, perfect. Thank you so
Okay,
much.
okay, I have no idea how that happened
Well,
because it's
technology can be
microphone.
funny. Thank you
Okay,
so
I'll
much.
continue on. Okay, okay, so as I mentioned previously, for each chromosome, there are 2 copies per cell. And one of those chromosomes came from the mother and another came from the father. Now, that means that you have 2 copies of every gene and 2 different ways that you can potentially make the proteins encoded by these genes. One driven by the mother's DNA, one driven by the father's DNA. Now, over many generations, the DNA, our DNA is not static. It undergoes minor changes over generations where the sequence of the DNA has become slightly altered, maybe due to some slight errors in copying. And as a result, over many generations, the DNA can become more and more different. And this can cause variations in the sequence of the DNA, which can then lead to variations in the proteins and how they function that are encoded by the DNA. So when we have a protein encoded by the DNA, that protein can be look exactly the same, like both copies of the genes, the one from the mother and the one from the father can be the same, or they can be slightly different, and they can have slightly different functions. And so the combination of these 2 proteins really determines how these proteins will function in the cell. And not every the gene doesn't always function exactly the same depending on what chromosome it came from and how different from the other gene the 2 copies are. Okay, so those differences we can test for. And then what we can do is tell whether the gene had one sequence or another sequence. And by testing for those genes and their sequences, we can see whether a particular gene with a particular sequence and a particular function for its protein is more often seen in a lupus patient than it's seen in a healthy control. And that might suggest that that gene functions to promote lupus. So for many years, we did this in what was called the candidate gene approach, meaning we would guess what genes might play an important role in the immune system and might be associated with lupus. And then we looked to see whether the function of these genes was different in lupus patients as compared to healthy controls. But then over time, we got a better and better idea of the sequence differences between DNA in the general population. And we learned how to type for these minor differences between DNA between individuals. And then using approaches where you can look at many, many genes at once, then we could start to do these studies that are called gene-wide association studies, where we could basically look at the variability in the genes throughout the whole chromosomes and the whole set of DNA. And what we could see is, where are there regions that are more often seen in lupus patients, certain sequences seen in lupus patients as compared to healthy controls? And if these regions of DNA differed significantly between lupus patients and healthy controls, it would suggest that in that region, there's a gene that could be associated with an increased risk of developing lupus. And so using this approach, which is called genome-wide association studies, or GWAS, a whole bunch of genes were identified that were associated with lupus, and we've learned a lot about the genetics of lupus. And one thing we've learned is that lupus isn't a simple disease where one gene causes the disease. So it's not like eye color. It's what's called a complex genetic disease, which means that many genes act together to promote the disease. And using genetic approaches, there are now over 300 genes that have been identified through GWAS and candidate gene approaches that are associated with lupus on various racial backgrounds. And what we've learned is that each of these genes really contributes a small increased risk to the development of lupus. So the odds ratio of developing lupus with the typical genes that are associated with lupus is about 1.1 to 1.5 fold over what's seen in the general population. So that's a very small increase. But for 80% of the genes that are associated with lupus, that's the kind of increase we're talking about. There are other 20% that are in the 1.5 fold to 3-fold increase in lupus, but anything over 3-fold increased risk is extremely rare. So the other thing is, these genes are prevalent in the general population, but by themselves, they're insufficient to cause disease. It's the combination of the genes that leads to the development of disease or increased susceptibility for disease, and then the interaction between these genes and the environment that causes disease. So each of these genes is thought to act independently, and they add together so that multiple genes are required to promote disease. So many of the genes, and this is from a study that we did a number of years ago that we participated in, many of the genes associated with lupus are not just lupus genes, but they're genes that are associated with the development of autoimmune disease. And here I think we have about 60 genes that were identified that were associated with lupus. And if you look, anything colored in blue on this inside wheel is a gene that's found in other autoimmune diseases, and in anywhere from 1 or 2 autoimmune diseases to 9 or 10 other autoimmune diseases. So many of the genes associated with lupus, and you can see the vast majority here, are actually autoimmune genes, not just lupus genes. And that brings me to the point that often the family members of lupus patients not only have an increased risk for lupus, but they also basically have an increased risk for other autoimmune diseases because these genes are autoimmune genes. And this is from a study we did many years ago, in which we were looking at looking at the family members of lupus patients, And what we can see is if you look at the first column, we had 628 lupus patients and we had 268 controls. And basically what you can see is that there's an increased proportion of the relatives of lupus patients that have autoimmune diseases. So increased rheumatoid arthritis, increased Sjogren's syndrome, increased hyperthyroidism, and hypothyroidism. So overall, we found in this study a fourfold increased risk in the general population. But I have to say that probably the risk is more like twofold for these autoimmune diseases, because the controls that were selected for this study were controls in which there was no history of any autoimmune disease. So the other thing that I want to point out is that most of the changes that affect the genes in lupus don't affect the actual protein itself. So the protein is the same. What it affects is how much of the protein is produced and when it is produced. So there's different regions of the chromosome of the gene that are responsible for this. So one part of the gene is responsible for encoding the protein, but other parts of the gene are responsible for controlling when that protein is expressed, how much of that protein there will be, and it's that part of the gene that tends to be different in these lupus risk alleles. So often there's either too much or too little, and it's being expressed at the wrong time. So what kind of genes cause lupus and how do they promote disease? So all of this work with all of these genes has led to the appreciation that the genes really fall into specific categories which allow us to determine something about the pathogenesis of lupus and the kind of genetic abnormalities that promote lupus. And there's basically three categories. The first is these genes can affect lymphocyte function, and I'll talk more about that. The second is these genes can affect the immune system's ability to sense danger in response to it. And the third is that some of the genes are involved in waste clearance. And these different categories of genes interact with each other to facilitate development of disease. So I think one thing we've learned over the last, say, 5 years with the advent of CD19 CAR T cells and how unbelievably effective they are in lupus is that B cells are central to the development of lupus, and the antibodies they produce are central to the development in lupus and damage to tissues. So the role of B cells really is to make antibodies. And the role of the immune system really is to protect us from dangerous threats in the environment. And things that endanger us are things like bacteria and viruses. And typically B cells bind to these bacteria and viruses. And with the help of another immune population called T cells, they change, they become activated and change to antibody producing cells, pouring out large amounts of antibodies. And these antibodies bind to bacteria and viruses, and their role is really to clear these bacteria and viruses from the circulation where they can be taken up and degraded and gotten rid of. Now, in lupus, not only does this normal function of B cells occurs, but also B cells recognize some self molecules. And they become activated in the response to these self molecules. And then they can differentiate into autoantibody producing cells. And then these autoantibodies can deposit in the tissue and they cause damage. And as I mentioned, with CD19 CAR T cells, we know if you get rid of the autoantibodies and you get rid of those bad B cells that are producing the antibodies, then you can treat the disease. So what kind of genes could then promote lupus? Well, the genes that promote lupus that affect B cells are basically ones that increase the ability of B cells to respond to their environment. So they basically deliver a much stronger signal to the B cell. Now, the thing that controls whether a B cell will respond just to foreign aid things like bacteria or viruses or to your self molecules within the body is actually how the B cell signals. And normally, B cells that recognize stuff in our body, which are our own self molecules, are inactivated. They're not typically gotten rid of, but they're inactive. And with the normal response to like foreign things, like a bacteria or virus, they don't get activated. But if you have um, proteins inside the B cell that enhance the B cell's ability to respond to these bacteria and viruses, then there's a stronger signal delivered to the B cell. And this can overcome this inactivation, and you can end up with B cells getting activated by self molecules. And that's all of the genes that affect B cell function basically act in this way to allow the B cells that are self-reactive to become activated. In addition to that, there's a whole group of genes that affect the T cells that basically provide help to the B cells to allow them to differentiate into these antibody producing cells. And the same kind of theme applies. These are genes that enhance the ability of T cells to respond. And because they have enhanced ability to respond, they end up responding to self molecules. There's more of these T cell helper cells. There's more of the T cells that can elicit inflammation in the tissues. And as a result, there's an increased response, which flows over to self. So as you have an increased response against your own self molecules. So the second group of genes that are associated with the development of lupus are those that affect the ability to sense danger. Now, the whole purpose of our immune system, as I mentioned, is to protect us from these outside threats. So along the lining of the barriers, so our barriers are our skin, there are the mucosa in our mouth, they're the inside of our intestinal tract. These are the barriers between the inside of us and the outside of us where there are potential threats to us. And so we have these innate immune cells that line these barrier sites, and their purpose is to recognize when there's a threat so we can activate the immune system so they can come and fight the threat. So whenever there's a barrier breached by viruses or bacteria, these cells sense danger. And the way they sense danger is by recognizing molecules that are commonly seen in these bacteria and viruses. And most relevant to the development of lupus is the fact that they do recognize viral RNA and DNA as well as bacterial DNA. And that is one of the danger signals that these innate immune cells respond to. So what happens is when these immune cells see this viral DNA, RNA, or other viral molecules or bacterial molecules, they sense a threat and they secrete inflammatory factors. And the purpose of that is to recruit immune cells to the site. And then they secrete factors that basically enhance the immune system's ability to fight infection. And one of those molecules is interferon. So interferon is a very important cytokine in lupus, and it's at elevated levels. And interferon plays a crucial role, for example, in fighting viral infections. And it does this by really affecting multiple immune populations and enhancing their ability to respond to the threat. So, for example, it increases the ability of B cells to become activated in differentiating to antibody producing cells. It expands the number of T cells that can provide help for the B cells. It allows the innate immune cells to present things to T cells to rev them up and activate them. So basically, the lupus genes, the lupus variants of the genes that affect this danger symptoms danger system are genes that actually enhance the ability to respond to danger signals, to produce more interferon, and to respond to interferon. And finally, the final class of genes that are associated with lupus are ones that are involved in waste clearance. Now, whenever the immune system is activated and revved up, all these cells expand and proliferate, and they're no longer needed after the immune system is finished getting rid of the threat. And similarly, the antibodies are binding to these bacteria and viruses. They're getting rid of the bacteria and viruses, but again, all of this stuff has to be gotten rid of, and we have to return to a more quiescent, relaxed state in terms of our immune system. And so there's a whole bunch of mechanisms to get rid of those excessive immune cells, to get rid of cells that are damaged or dying. And one of the ways that this occurs is through this process called apoptosis, which is also called programmed cell death. And the thing about apoptotic cells is normally they're very rapidly cleared from the bloodstream and from the tissues so that they don't persist. And as a result, the immune system never really sees them. However, if you look at these apoptotic cells, these cells that the immune system's getting rid of, they actually have nuclear proteins, RNA, and DNA on their surface. These are all the things that are recognized in lupus. And as I said, if these cells or bits of the surface of these cells persist in the bloodstream and or tissues, what can happen is these DNAs and RNAs and the proteins on the surface can signal danger to the immune system. So this is where the danger system is invoked. They can signal danger to the immune system and they can elicit an immune response. And then because you have these lymphocytes that are capable of reacting to self-proteins, they can be recruited and they can respond. So gene changes that impair the ability to clear these cells or the bits of cells or impair, the body has many ways to degrade RNA and DNA because they do constitute danger signals. So anything that impairs the ability to degrade RNA and DNA tend to promote lupus. And similarly, gene changes that impair the ability to clear all those antibody molecules and that are bound to things will tend to promote lupus because they enhance the ability of these complexes to bind to the tissues and to elicit inflammation. So what is I'm going to leave the genetics a little now and move on to the role of the environment. So there's a whole bunch of potential factors that could affect the development of lupus and that have been proposed to promote or increase the risk for development of lupus. And in particular, bacteria and viral challenges, vaccinations have been proposed, smoking and particulate matter in pollution. There's a role proposed for hormones. I'm going to talk a bit about diet. We all know that, sun exposure can lead to rashes and sometimes flares of disease. And then there's some chemical exposures. So one of the things that's thought to play a relatively important role in perhaps affecting the risk for lupus are viruses. And so there's a lot of work that has been done over the years looking at, can lupus onset after specific viral infections? Do viral infections promote flares of disease? And people have looked at many viruses. And there are publications proposing that certain viruses play a role, and other publications showing that they don't play a role. And part of the problem here is that many viral infections promote an illness that can look very much like lupus. So it's hard to tell whether this is the first sign of lupus or whether the symptoms are related to the virus or not. But the three examples I've shown here are really examples where there's a fair amount of data to suggest that these specific viral infections may be associated with either flares or an increased risk of developing lupus. And I do have to point out that it may not just be one viral infection. It just may be all these challenges by bacteria and viruses that eventually lead to promoting the development of lupus. So there's quite a bit of data on Epstein-Barr virus, the virus that causes mononucleosis. And it's based on the fact that the antibodies that demonstrate that there has been an infection with Epstein-Barr virus are present in more lupus patients than in healthy controls. And in addition, the types of antibodies differ a little bit in what's seen in lupus patients as compared to healthy controls, in that they're more the antibodies that indicate, so this virus kind of hides in the cells and every once in a while can come out and kind of reactivate. And so the antibodies that are seen more commonly in lupus patients are the ones associated with viral reactivation. So there is data, pretty good data, suggesting that the infection itself is not associated with an increased risk of lupus. What I mean is the primary infection. Having mono, for example, is not, that does not immediately impose a risk with developing lupus. It more appears that the virus is there for a prolonged period of time, and then at some point it reactivates, and that then might subsequently lead to the development of lupus. And there is data suggesting, for example, that ANA positive individuals who have high levels of these antibodies suggesting the virus might have reactivated are more likely to go on to develop lupus than those that don't. In the case of influenza, recent infections are associated with an increased risk of flare, but the data suggesting that an influenza infection subsequently results in the development of the new onset of lupus really is not as compelling. And more recently with COVID, and this I have to say is an evolving story, COVID infection itself, like an acute COVID infection, is associated with high levels of anti-nuclear antibodies in up to 50% of the patients. And there are a few cases of SLE developing during the acute phase of infection. But as I said, because this is a new infection, it's really not known how much of a role it will play as a potential trigger. So in contrast to viral infections themselves, there really is very little evidence that vaccinations promote the development of lupus. So in fact, our traditional vaccinations for influenza, for diphtheria, pertussis, tetanus, etc., all of those, it's clearly being shown, are not associated with an increased risk of developing lupus after vaccination. So they've looked for at least 24 months after a vaccination, there's no increased risk. Vaccination for HPV is associated with a localized sort of skin reaction that can look like lupus, but there are no cases of systemic lupus developing after HPV immunization. There are a few cases, though, of lupus onsetting 1 to 2 weeks after COVID, and there are several case reports of flares developing after vaccination with COVID. But this is an evolving story, and it's unclear what the prevalence will be. But overall, you know, given the very weak data that lupus risk is invoked by giving vaccinations, it is not recommended that vaccines be held to minimize lupus risk. So an evolving and really exciting story is the role of the gut microbiome in potentially promoting the development of autoimmune diseases, including lupus. So the gut microbiome is basically a whole collection of organisms that live inside the human intestinal tract. And these organisms actually have good and bad roles, but overall, they're there all the time. They're compatible with health. The organisms themselves secrete factors that help to maintain the gut wall in a healthy way, so it makes an effective barrier. And not only that, many of the organisms secrete factors that basically serve to dampen down the immune system, so we're not always trying to fight these bacteria that are normally there and provide a healthy role in the gut. So there is evidence in lupus and other autoimmune conditions that the collection of organisms in the gut is different than in healthy controls. So there's a whole bunch of these different bacteria, and so it's a less diverse collection of bacteria in patients usually with autoimmune disease as well as lupus patients. And there's an imbalance in the types of bacteria which are present. And so in general, the bacteria that basically maintain gut integrity and have a kind of healthy immunosuppressive or anti-inflammatory role in the gut are decreased, and those that are cause more inflammation are increased. So there's this concept that because there's more of the pro-inflammatory bacteria compared to anti-inflammatory bacteria, that the wall of the intestine is not as healthy and that you get a leaky gut. And that leaky gut may promote the development of autoimmune diseases. So there are several studies that suggest that the gut is leaky in lupus. Now, it's not entirely clear when this occurs. Does this occur at the onset of lupus, or does this occur at the onset of anti-nuclear antibody development? Because you can develop anti-nuclear antibodies years in advance of developing lupus, and many people that have anti-nuclear antibodies, in fact, 20% of women have anti-nuclear antibodies, never develop lupus or an autoimmune disease. So it's not entirely clear, but definitely lupus patients have evidence of a leaky gut. And there is some evidence from very limited number of studies that the microbiome in people who are just ANA positive may also be shows changes somewhat similar to what you see in lupus. and that there's at least one paper that suggests that those changes are associated with possible leaky gut. So what does a leaky gut do? Well, it allows bacteria and bacterial products to get through the gut wall and to activate the immune system. So these bacterial products sort of get through the gut wall. They signal danger to the cells that are underneath the gut wall. And then the bacteria can elicit an inflammatory response. So now, and that occurs it is thought to occur as playing an inciting role in many autoimmune diseases, including rheumatoid arthritis. So the data suggests, though, that in lupus, these bacteria also have nuclear antigens. And the bacteria nuclear antigens look very much like our own nuclear antigens. So that if you have a defect that allows you to respond to self-nuclear antigens or nuclear proteins and DNA and RNA, then these bacteria can actually elicit these anti-nuclear antibodies, and they can activate anti-nuclear antibodies. Now, this doesn't normally happen in a normally functioning immune system, but if you have, as I said, a defect or a change in your immune system that allows you to respond to self-nuclear proteins and DNA and RNA, then these bacteria can activate them. So then that brings us to the potential role of diet, because diet actually has been shown to play a very important role in modifying the microbiome. So there's a couple of pieces of evidence suggesting that diet is important. So there was a nurses' health study in the US where they studied 240,000 nurses. And what they found was that nurses who had a high intake of ultra-processed food had an increased risk of developing lupus with an odds ratio of 1.5. And similarly, in the Black Women's Health Study, a diet high in carbohydrates was associated with an increased risk of developing lupus. So there are two kinds of diets that have disparate effects on the gut and the immune system. So if we look at a diet that's rich in fiber, so this is vegetables and fruits, something like the Mediterranean diet. And if you look at that diet actually promotes the kind of bacteria that produce this factor called small chain fatty acids. So these small chain fatty acids promote gut health. They actually make the lining of the gut stronger, and they make it strong so that it's not leaky. And they also play a role in impairing the immune response to these gut bacteria. So preventing an abnormal response. So that diet is good. If we look at the diets that are high in carbohydrates and fat, the converse happens. This actually promotes bacteria that damage the mucosal barrier that kind of helps to protect the gut. And these bacteria can invade the gut can get leaky, and the bacteria can get through the bacterial wall, and underneath they can elicit a pro-inflammatory response. Not only that, but diets that are high in carbohydrates and fats promote obesity, which is also associated with a pro-inflammatory state. So I know I'm running late because of some technical difficulties, but just a couple of other points before I finish up. So treatment with exogenous hormones is associated with an increased risk of developing lupus. So there are two really nice studies, one from the UK, another one from the Nurses' Health Study, that show that use of oral contraceptives, or in the case of the Nurses' Health Study, administration of postmenopausal hormones is associated with an increased risk of developing lupus. And this kind of makes sense because we actually know that estrogen has pretty profound effects on the immune system. It does serve to enhance B cell activation. It's known that estrogen promotes the development of B cells that can react to our own self molecules to a greater extent. And also, it's known that estrogen will enhance the ability of the body to these innate immune cells to respond to some danger signals. UV light, as you know, is also a well-recognized factor in lupus and is associated with photosensitive rash, and it can induce flares. There actually is no definitive evidence that UV light, more or less, it would be hard to quantify, I think, is associated with an increased risk of lupus. But there was a study that looked at ANA positive individuals who were at risk of potentially developing lupus or other systemic autoimmune diseases. And when they biopsied their skin, those patients that had high levels of interferon in the skin were more likely to progress over the next year than those without. So that suggests that some kind of maybe abnormal or different response to sun exposure might be associated with an increased risk of developing lupus. And I think I'll just move on. I will say that a variety of respiratory exposures are associated with increased risk of developing lupus, silica dust, smoking, and in the case of smoking, it's current smoking. So if you're a current smoker, you have an increased risk of about 1.5-fold to develop lupus. And then there's a number of studies basically showing that air pollution is associated with an increased risk of lupus. And I'm not going to talk about chemical exposures, but there are a number of chemical exposures that are reported to be risk factors for the development of lupus. Okay, so I'm going to end before I summarize by talking about the Nurses' Health Study and what it's kind of told us about the interaction between genetics and environment in the development of lupus. Again, they studied 240,000 nurses. They gave them a bunch of questionnaires so they could ask them about their lifestyle, their reproductive and environmental exposures, their medical history. And of all those nurses, during the period of time they were studied, 138 developed lupus. A number of them were genotyped. And when they genotyped them, they looked for lupus genes. And the more lupus genes, they calculated a score based on how many lupus genes you had. And the more you had, the higher the risk score. And what they found was if you had a higher genetic risk score and a family history of lupus or rheumatoid arthritis, you were more likely to be one of those individuals that developed lupus. And then they looked at the environmental exposures that were associated with an increased risk of lupus. And basically, they found that cigarette smoking was associated with an increased risk, compatible with other studies. Low alcohol consumption, the reason for this is not entirely clear. Obesity, as I mentioned, this is associated with a pro-inflammatory state. Early menarche, I guess more hormones at an earlier phase of life, and oral contraceptive use. So in summary, studies provide evidence for a clear role for genetic factors in the development of lupus, and ongoing work has provided insight into the types of changes in gene function that are associated with development of lupus. However, given the kind of complexity and number of genes, we're not able yet to determine with any great degree of accuracy the risk of a single individual developing lupus. For example, we can't genotype the whole genome and say, based on this genome, there's this risk of developing lupus, because there's no one gene and we don't understand how they all interact with each other to promote disease. There are a number of environmental factors that have been shown to influence the development of lupus, but I think a lot more study is required to be able to translate what we know about environmental factors, i.e., what we know about the microbiome and diet and its role, into accurate prediction of risk, and also into how we could strategize to use some of this knowledge to develop preventative therapies. So, questions? I know.
Thank you, Dr. Wither, for that wonderful presentation. I certainly learned a lot, especially on the genetics, especially diet. That's super interesting. But yeah, just for the sake of time, I'll quickly turn to the chat. Feel free to post any questions that you may have, and I'll filter through. The first one being, would stress fall under environmental?
Yeah, so I think stress would definitely fall under environmental influences, and we know that stress impacts the immune system. So I would definitely say that. I don't know of any specific studies that have looked at stress and its impact on the risk of developing lupus, but I wouldn't be surprised if it doesn't. If it does, or I'm using two double negatives, but basically what I mean is that I would think that it does play a role.
Okay. The next question, since there's no Epstein-Barr virus vaccine, is there a way to avoid this infection, or do we just do our usual precautions we take as immunocompromised people?
Yeah, that's a good question, because it's endemic and we get it. Just use your usual precautions. I don't think there's any real way to avoid an Epstein-Barr virus infection. I think we just need to understand better what leads to its reactivation in certain circumstances, and to prevent that.
And are exogenous hormones, for example, birth control, a factor in flares for women who already have SLE?
Okay, so I did read about this, and so in terms of taking exogenous birth control pill, for example, if your disease is inactive and you don't have antiphospholipid antibodies, because hormones are associated with an increased risk of thrombosis, then there isn't, then it's not, as I understand, it's not it's not contraindicated, and it poses a minimal risk.
And another one.
That's what I read, but I'm not I really, unfortunately, am not an expert, but this is what I recall reading.
And another one from the Q&A. Are there any protocols or supplements for helping repair the gut barrier?
So, it is, I would say, because I'm often asked this question, that a Mediterranean diet or a diet high in fiber will help to promote the kind of bacteria that are associated with repair of the gut.
Right.
But to date, there's no definitive evidence that that particular gut will, it's not, how can I say, it's not going to treat lupus, but it might help to attenuate the issues related to the gut microbiome.
Okay. And another question, is a positive ANA and mouth ulcers on the lip and cheeks a confirmation of lupus?
No. The
Okay.
answer is no. Um, you know, to make a diagnosis of lupus, it really requires, could it potentially be associated with lupus? Yes, but that, you would have to look at the constellation of other features that were associated with those two things.
And just a few left. Are antibiotics a culprit as well?
Antibiotics can disrupt the microbiome. Yes, they can lead to some disruption.
And for the population of people with lupus that don't test positive for any of the anti-Smith or specific antibody tests, but a very high ANA, what is the cause for this?
Um, I think basically the same mechanisms are involved. Like we study women that just a whole variety of women that have positive ANAs. Some of which have specific antibodies and other which don't. And immunologically, they look very similar. So I think the same mechanisms are involved in developing an ANA. I think there's a little added complexity with the type of antibodies you produce, which has something to do with certain changes in the immune system. But I think fundamentally what produces an ANA is very similar amongst all ANA positive women.
Thank you so much for answering those questions, Doctor. Whether I know there are a few others in the chat. Maybe if you just want to look, maybe you could answer those directly. But just for the sake of time, we do have to move forward to the next